Good morning, and we were saying CSIR Net Life Science memory based question. One of the question was which of the following is correct for cleaving DNA? Options were Cas, Cas9, DCas, and Cas-JB. So the natural origin of Cas9 protein has come from prokaryotes, isn't it? Prokaryotes like bacteria and archaea, they have this Cas9 protein, and when a bacteriophage like virus infects them, then the Cas9 protein cuts the RNA particle of the bacteriophage, isn't it? And it attaches that RNA particle into onto its own DNA and remembers that this uh, bacteriophage has infected me, and in future when the same bacteriophage infects the bacteria, then it can rapidly uh, make defense activities against that bacteriophage, somewhat like adaptive immune system that is present in humans, isn't it? So you can see here blue, orange, red, and green, and then again red structures like this is what is the various RNAs of the bacteriophage, which the bacteria has been assembled and attached in its own DNA, isn't it? So Cas9 is a protein which occurs naturally as defense system in the bacteria. So this protein is being used in CRISPR, isn't it? And CRISPR-Cas9 gene editing steps. So this in CRISPR, what the steps is, we first of all in CRISPR we find a target RNA and target DNA. Sorry, we find the target DNA to be cleaved, isn't it? So let's say here you can see, and in response to that target DNA, we make a guide RNA. First of all, the first step is to design the guide RNA. Here you can see GUCAA-GU. So in opposed to this RNA, guide RNA, there will be a sequence in the genome in either in humans or in mice or any other host organism in which the CRISPR-Cas9 gene editing system is to be used. So this guide RNA is then associated with that Cas9 protein which has been originated from the bacteria. So these both are combined, and this is known as CRISPR complex. And this Cas9 complex is then introduced into the host cell, isn't it, by various methods like gene gun or electroporation or any other method, isn't it? So then that CRISPR Cas9 complex uh, surveys in on the whole DNA, isn't it? It searches for that particular strand and then makes the cut at that particular sequence that has been made in opposed to the guide RNA. So after that cut is made, there can be two fates of the genome, isn't it? So a direct repair takes place, homologous recombination takes place due to the damage of the DNA, and the gene and the genes is repaired. Uh, and what the gene has, what the CRISPR Cas9 has cut, it has already gone. So in absence of that, it is known as knockout. Okay, and when a new strand along with Cas9 is added, isn't it? It carries the DNA part that has to be added in the gene in the DNA. In that case, it is known as knock in. Okay, so now you know how the CRISPR Cas9 works. These are the literature. First, the guide RNA is designed in opposed to the targeted DNA. Then it is formed complexed with the Cas9 enzyme. Okay, the guide RNA and Cas9 enzyme is complexed, and then it is inserted into the cell, and then the after searching, the CRISPR-Cas9 cuts the targeted DNA, and then after the homologous recombination or the repair enzyme takes action for the subsequent cut, isn't it? So this is what CRISPR is. With the help of CRISPR, there is genetic knockout mouse is being produced, or knock-in mouse will be produced. In this, what happens, the stem cells are taken out of the embryonic, a pregnant mouse is taken, and from that pregnant mouse, the what the blastula stage cell is there. From that blastula stage cell, the embryonic stem cell is taken out, and in that stem cell, the CRISPR Cas9 protein is inserted, and then the gene edited cell is again replaced in the blastocyst, isn't it, by various methods like injection. It may be gene gun or it may be injection, whatsoever the method be, and then that blastocyst, edited blastocyst cell, is placed inside the pregnant mother, isn't it? And when the babies are created, it is either knockout or the knock-in mouse, isn't it? So this is the same image. And in 2018, Lulu and Nana were two HIV-resistant CRISPR babies produced by Chinese scientist He Jiankui, and who claims to have created first genetically edited babies. And many other patients across the world have been performing clinical trials on humans for treating various cancer, immunotherapy, ophthalmic diseases, and metabolic conditions. You can search these names across the web. And option A, coming on to the question, option A was the Cas, isn't it? So Cas is not a DNA cleaver. It is a family of protein that cleaves the DNA. Okay, so certain only Cas is not a correct option. So Cas9 is what is correct, and Cas9 is the correct option that performs the function of DNA cleavage. And DCas is the dead Cas9. Deactivated Cas9 is known as DCas. So this is also not correct. And Casgv is the therapeutic product that is created for, so as seen here in sickle cell anemia. Let's say, for if you are diagnosed with sickle cell anemia, then you go for the CRISPR associated medicinal therapeutic, isn't it? So in Casgv, what is done, you evaluate, and the in a clinic, your stem cell is being collected, and the stem cell is undergoing the Cas9 complex gene editing, and the production of gene edited cell is again collected. And after that, third step is what, your whole whole hematopoietic stem cells that are present in your bone marrow, which we are going to form the red blood cell, is flushed out, and the new CRISPR edited cells are then replaced with that original hematopoietic stem cell that you have originally have. And then there is a recovery process in which you are monitored, and this whole process costs you around 20 to 23 crore. You can search this image. Thank you, and stay tuned for more questions.